5:32. I think I'll go ahead and kick off our event. So hello from Berkeley Public Library. Thank you so much for tuning in tonight. We are so excited about our partnership with Popping the Science Bubble. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create some constructive discussion about a variety of science topics. We have two speakers at each seminar who will talk about their current research or a topic that they find just absolutely fascinating. The organizers are three graduate students at UC Berkeley, Jenna, Madison, and Oksana. If you're interested in checking out their past seminars, you can visit their website, check out their Twitter or Facebook, and sign up for the listserv to keep in the loop for upcoming seminars. Our speakers are absolutely open to answering questions. So please do ask questions in the chat. Thank you so much for attending. All right. I will let our Popping the Science Bubble leaders introduce tonight's speakers.
All right, thanks, Kelsey. And we just want to emphasize that feel free to ask questions during the presentations. We'll be monitoring the chats on both Zoom. You can ask in the chat box or the Q&A box, as well as Facebook Live once we get that up and running. And we'll ask the speakers your questions in real time as time permits. And if we can't get to your question in the moment, or if you have additional questions after their talks, we'll have time at the end as well. And so with that, we'll get started.
Our first speaker of the day is Priya Garg, who was born and grew up in the northern state of India called Punjab. She is pursuing a PhD in the Department of Mechanical Engineering at UC Berkeley in the Fire Research Laboratory. Before that, she completed her aerospace engineering degree in India and her master's in space engineering in Italy. Her research is based on understanding the health consequences of wildland fire smoke on firefighters. Outside research, she enjoys hiking, listening to music, and cooking. Take it away, Priya.
I'll share my screen and I hope it's possible. Okay, can you see the screen, right? Slides?
Yes.
Okay. Okay, thank you so much for the lovely introduction, Oksana. So tonight, I'm going to briefly discuss the effect of moisture content and fuel type on emissions. Over here, I'm talking about emissions that is actually the smoke that is released during the fire season. And I'm pretty sure that everyone has experienced it. But before I get into my topic, like my research topic, I would like to give a brief introduction, background on wildfires, and then discuss my research. So to have a wildfire, or in general any fire, we need three basic ingredients: oxygen, fuel, and heat. And combination of these three is called fire triangle. And in addition to it, we also need a chemical reaction taking place. So four of them together is called fire tetrahedron. And out of these four components, if I take away any one component, either oxygen, fuel, heat, or like chemical reaction, we will not have a fire. And once we have a fire, then the behavior of the fire is influenced by many factors, and which includes climate, topography, and weather. And combination of these three is called like fire behavior triangle. And when we talk about like weather conditions like wind, which we are even seeing today. So if we have like very high winds, so the spread will be really fast, like fire will spread really fast. And similarly, higher temperatures, that means fuel in the forest will catch the fire like very quickly. And when we talk about fuel, so how densely are the forests packed? How densely are the forests packed? Like, are they like really dense, or there's like space in between? Will the spread happen from one tree to another tree? So things like that are considered in under fuel. And when we talk about topography, so if we have a fire that is starting at the bottom of the hill, it will very quickly spread to the top of the hill. And it's because of like the slope, topography, and also like the wind conditions affected. So these are the two fundamental triangles that determine the ignition and behavior of the wildfire. So over the years, we have seen that the number of wildfires is somewhat steady. That's this blue line. But the size of the fire has increased over the years, and warm climate has played a major role in this increase. So as the area is increasing, so does the suppression cost. So on the right, I have the plot of the suppression cost that has increased over the years. And because this area is increasing, so wildfire is a major cause for the destruction of property and loss of life. But additionally, the smoke causes like very serious long and short-term health effects. So that's what like we study in the lab. And what are actually these emissions? So what is smoke composed of, and how do we perform the study in a lab? That's what like I'll be discussing in this slide. So wildfire is actually a mixture of gases, particulate matter, and even metals. So particulate matter, this PM, is nothing. It's just like the small particles that you must have seen getting deposited on the cars and stuff in your like house. So that's like small particulate matter. And the very small size of particulate matter, like below 2.5 micrometer, gets deposited even in our lungs. So that's like a major health hazard. And I'm sorry. Yeah, so how to study the health effects is the thing that we are looking into. So one way to study the health effect is to track the health of local population. This is hard for wildland firefighters because they work seasonally. We can still pursue the short-term studies on the wildland firefighters, but long-term studies are not possible. And the long-term studies have been performed on structural firefighters that deals with fire in the buildings, and it showed increase in lungs and heart-related diseases. So how are these studies performed in the lab? So usually like in the lab or like in the research field, animal models are commonly used for tobacco and vaping studies. And that is one of the way to pursue wildfire smoke effect study. And presently, our collaborators at Northeastern University are pursuing the study using mice model. So they have like mice that's exposed to the smoke, and they study like how their lungs and heart are affected and correlate the results to the humans. So how do we actually characterize the emissions? Like how do you produce smoke in a lab scale or like field scale? So basically we can characterize the emissions like at different scales. So this is a field scale in which you just burn a piece of land and record the emissions. But obviously this is like the most realistic case you can have, but obviously there are like some drawbacks, and I'll be discussing that next. And secondly, we can characterize the emissions at the lab scale. You can have like experimental setup in the lab and characterize the emissions. This is actually my supervisor. He's trying to understand the effect of slope and wind on fire. And lastly, we can have a small scale setup to have a steady state smoke produced for long duration of time. So these are actually the three different ways to characterize the emission. And in literature, people have performed studies using all the three methods. But obviously they all have their own drawbacks, which is what I'm going to discuss next. So in terms of real representation, like what is the reality of the emissions? Obviously field scale does give you the reality, like real essence of what the emissions is. Lab scale somewhat give it, small scale does not. But in field scale, it's very difficult to place the instruments that record the emissions. It's very difficult at field scale, but it's really easy in the lab and small scale experiments. Okay, yeah. So, and the other thing is like, if you want to understand the emission, like how does emission change with change in fuel type and moisture content, that's very difficult at field scale because in field you have like fuel ranging from like different varieties to different moisture content. Like there's fuel that is completely live and completely dead. So you cannot have the differences in a field scale, but it's possible in a small scale experiments. And then the combustion condition. So basically we study about two different combustion conditions. One is flaming and one is smoldering. Flaming, when you actually see the flame, so this is actually the flaming experiment. And smoldering, you know, like when flame is gone, at the end you just have like smoke coming out. That's called the smoldering condition. And in the field scale, like when you burn a piece of land, all of these like different combustion modes happens at the same time. So you cannot distinguish if you are recording the emissions from flaming or smoldering. But again, this is possible at a small scale experiment. And lastly, we want steady state of emissions. Like when firefighters are exposed to smoke, they're exposed for a longer duration of time. And you want like the steady state smoke for long duration. And that is again possible in the small scale experiment. So out of these three scales, we chose to pursue the study with small scale. But the only problem that we have is the real representation. But if we correlate our results to the field scale, we can somehow like agree that, okay, this is correct. Like there's nothing wrong in the way we characterize the emissions. So, okay, so this is what the experimental setup looks like. So in this, I have a heater. That heater moves on the top of the quartz tube. So this is like a transparent glass quartz tube. And inside this quartz tube, I have a quartz boat that holds the fuel. So according to that fire triangle, you need heat, that is from the heater, you have a fuel, and then you need oxygen, that is like from the air. So air goes from like right to left and heater moves from left to right. So the fuel burns and we have emissions like smoke generated. And before that smoke goes to the exhaust, we dilute it with the air so that, you know, the instruments that we place does not saturate. So we have to take care about the instruments. So yeah, so these dotted lines are actually the sampling lines that are connected with three different instruments. We have NRAC that record the emissions like gaseous emissions, carbon dioxide, carbon monoxide, oxygen, sulfur dioxide, nitrogen oxides, and hydrocarbon. And then we have dust track. It provides you the real-time measurement of particulate matter. And then we have a filter that gives you the time averaged particulate matter, like the total particulate matter that is that we get through from the experiment. And yeah, this setup is based on a German apparatus that's DIN 53436. And this is what the setup really looks like in the lab. So in the lab, this white is actually the heater that moves on the outside of this quartz tube. And this heater is mounted on a traverse so that it can like move forth and back. And on the end of this quartz tube, we have an air inlet. And this is the power supply that controls the temperature of the heater. So this is what the experimental setup is. And over here, like inside this quartz tube, we place the quartz boat with the fuel. Now, what is this actually fuel? So I have like performed study with 4 different fuels. Where Douglas fir and lodgepole pine were shipped from Missoula, and pitch pine and mountain laurel were shipped from New Jersey. And these are the fuel that are actually studied as wildland fuel in the literature. Okay, yeah. And to understand the effect of moisture content, because you see these are like pretty green. So to understand the effect of moisture content, I have to like decide 3 different moisture levels. One is like when they're completely green, that's like they're live, but they're like plucked from the tree. So they're like recently live. One is like completely dead. And then one is like intermediate state, which I called as wet. So these are like 3 different moisture levels that I selected. And moisture content, it's nothing, it's just like the mass of water in the sample divided by the wet weight of the sample. That's how we define the fuel moisture content. And on the right, I have 2 images, smoldering and flaming. So in the flaming test, you see this small flame, and this white is actually the heater. It's actually insulation on the top of the heater. In these tests, you will see the flame continuously for 1 hour of experiment. So this flame will be there continuously. And then we have the smoldering experiments in which there's no flaming, but there's like a little bit of glowing, and you have the smoke produced continuously for like 1 hour. And the good thing about these experiments is the amount of fuel that we use in this like quartz board, it's very less. It's like less than 15 grams. And now moving on to some results.
Oh, Priya,
Yeah.
I had a question, maybe just a very general one. Why did you choose these different types of trees? Like, yeah, fir, pine, like how did you all choose them?
Okay, so because I showed you like small scale experiments needs to be related with the field scale experiments. So in the literature, you have a lot of data available for these fields. So I have to choose for which like I already have results. And we had like a pretty steady supply from the people like Missoula Labs and like Pine Barrens. So like somebody have to go pluck it and, you know, ship it overnight. So we had like a pretty steady supply also. So that's the reason we chose these 4 fuel types.
Okay, thank you.
Yeah. Okay, yeah. So moving on to the results. So this is like the time, over the time, what emissions look like. So this blue line is actually total particulate matter. And then we have hydrocarbons, carbon dioxide, nitrogen oxides, and emissions are like pretty steady over the time. That was like the aim of the small scale setup, but we see little increase in carbon monoxide, but that's all right. And this is actually flaming Douglas fir at the intermediate. It's not like completely live or dead. It's like wet state with 10% moisture. And on the right, I have a plot that shows carbon monoxide emissions for flaming and smoldering for 3 different conditions, like live, that is green, dry, completely dead, and wet is the intermediate state. So these dotted lines are like smoldering experiments, and solid lines are flaming experiments. So what we see for the smoldering experiment, as the fuel moisture content goes to 0, as the fuel moisture content decreases, we see increase in carbon monoxide production. So that's what we see in this dashed line. Whereas for flaming, for completely live and dead, carbon monoxide production is high, whereas for like intermediate state, carbon monoxide production is less. So we see like emissions does change with change in the moisture content and combustion condition. So that's what we found out. And now, like, before I move into more results, I just want to give like 2 definitions. One is emission factor, like definitions that are commonly used in wildfire research. Emission factor is defined as the mass of any species that you are looking for, like mass of carbon monoxide in grams divided by dry mass of the fuel. So that's how emission factor is defined. And in order to compare the results with the literature, I like calculated the emission factor for all the gaseous and total particulate matter species and plotted it on a graph. And okay, this is flaming emission factors for Douglas fir. So over here we have total particulate matter, carbon monoxide, carbon dioxide, and I just like divided it by 100 and 1000 to have all of them on one plot, and nitrogen oxides and sulfur dioxide. So what we actually see is, as again, like as the moisture content changes, we see change in the total particulate matter production. So there is like, it increases from live to wet and then decreases from wet to dry. Same for carbon monoxide, we see like huge decrease from live to wet and then increase from wet to dry. Carbon monoxide and and nitrogen oxides, they're decreasing with decrease in moisture content, and very small contribution from sulfur dioxide. Over here, I have not plotted hydrocarbons because they were negligible for flaming experiments.
Priya, can I ask a question?
Yes.
It seems like the difference between live, wet, and dry, or as they've been defined, primarily would be moisture content. Do you have any idea where these carbon species are coming from that are so different between the samples?
We have like carbon in the fuel itself, right? Fuel is like, you know, the fuel is composed of carbon, nitrogen, and sulfur, like the basic components that we have, elemental components, and nitrogen, elemental components of the fuel. So fuel does have carbon in itself. Does that answer the question?
Yeah, I think I was a little bit, I guess my question, maybe I didn't phrase it right initially, was more of, I guess, yes, the carbon-based compounds are coming from the fuel itself, but what makes it so much more emitted when a fuel is live or has a higher moisture content?
We really don't know. I think, like there are like different volatiles because the liquid is not actually water in the fuel. And we did not like completely dry it and then, re-wet it with the water. We dried it on a very low temperature so that some volatiles are preserved, or like some resins in the leaves or like needles are preserved. And probably that's those volatiles are changing the emissions. That's what we assume.
Cool, thanks.
Yeah. Okay, moving on to the smoldering results. So smoldering, when we don't really see the flame, and we see that the variation is pretty less compared to what we see in the flaming. But the major component that we see for the smoldering is actually hydrocarbons. So not completely burnt hydrocarbons. So we have a huge amount of hydrocarbons that is produced for smoldering emission factors. So if I compare the results of smoldering and flaming, so this is like Douglas fir, smoldering, flaming at 3 different moisture levels. So we need to see that the amount of total particulate matter that is produced for smoldering experiments is like 50 grams per kg of dry fuel burn, whereas emission factor for flaming, it's like less than 1. So that's like a major difference between smoldering and flaming experiments. And then if you see this hydrocarbons, you see like there's like huge amount of hydrocarbons, probably around like 25 or like 30, but hydrocarbons for flaming were negligible. So I didn't even plot it that. So these are like very major differences that we see. And we also see like change in terms of the moisture content. As you change the moisture content of the fuel, you change the emissions. And using these plots, like I have shown the results only for Douglas fir, but I did perform the experiments for lodgepole pine, mountain loria, and pitch pine. We went with the extreme case, and that was actually smoldering Douglas fir. So smoldering Douglas fir has the highest amount of total particulate matter released. And this bar is the standard deviation among the experiments that I performed. And we thought of exposing the mice to this condition, dead smoldering Douglas fir. So moving on to another definition.
Priya, yeah. I was just going to comment, that's really cool, because I guess when I would think of a smoldering fire versus like a live fire, that the live fire would have more particulate matter for whatever reason. But I guess in practical terms then, say like, we're all thinking about N95 masks right now. Would it be fair to say that it's more important to have an N95 mask on if there's a smoldering fire around because there's a larger amount of particulate matter?
Yes, that's correct. For smoldering, we need N95. And that's actually a part of my research also. How good are these N95s? So that's the next part in my research. I'll briefly discuss about that also.
That's really cool. Thanks.
Yeah. So,
Priya, sorry, if I might also ask, I mean, the way that you apply like these results in terms of firefighters obviously are looking at long-term effects, but firefighters, are they spending more time in I would assume they're spending more time in live fire zones than smoldering fire zones. And do you guys then try to like minimize time spent in smoldering zones post? I guess what would that translate to when trying to inform like how firefighters would get exposed to wildfires?
Okay, so firstly, I think in the field they get exposed to the combination of flaming and smoldering. It's not only one condition. But when we study about the health effects, we prefer to go with the extreme situation. Like obviously, like you are getting exposed to, let's say, 10 grams per kg of total particulate matter, but it's better to study for 15 and then see like how are they getting affected, how lungs and heart are getting affected. And in terms of correlating, so at Northeastern, they have a big matrix of correlation, which I personally don't understand because it's something related to bioengineers. But they came out to have like exposure for 2 hours every day for the mice in order to see like a day exposure for the human beings. So that's a kind of correlation they have.
Yeah. Great. Thank you. We also have a follow-up question in the Q&A if you have time for one more, from Joseph Heisler, who asks, was the intent of your experiment to compare relative differences in emissions for different fuel types rather than estimate actual exposure concentrations to firefighters or the public?
So actually it's both. So the first part, like relative differences in the emission factor, that's what I do. But from that, we concluded, okay, this is the extreme case. And only the extreme case scenario is used for correlating with the firefighters. All these scenarios are not related to like mice study and humans. No, only one of the situation is getting correlated right now.
Great, thanks.
Okay, so another definition is modified combustion efficiency. I'll actually go a bit fast. So it's actually the ratio of amount of carbon dioxide divided by the sum of carbon dioxide and carbon monoxide. And we see that the amount of carbon dioxide released in flaming is pretty high because it's a kind of complete combustion. So if the modified combustion efficiency is more than 0.95, it's actually flaming combustion. And if it's between 0.65 to 0.85, it's smoldering combustion. And we have like these two basic definitions to correlate the results from small scale to the field scale. So this is what the comparison looks like from the literature. So I compared my results, these green triangles with the literature. On the x-axis, I have modified combustion efficiency, and on the y-axis, emission factor for carbon dioxide. And I compared my results with two field studies, like burning a patch of land, two lab studies, and one small scale studies. And we see like we are pretty much in a good agreement. And this is for emission factor of CO2. We did same for emission factor for carbon monoxide and for particulate matter. And yeah, so this is the study that have been done at Berkeley, like by Fire Research Lab at Berkeley. And moving on to a little bit of the mouse exposure chamber that I have been talking about, like the study what is happening at Northeastern University. So we duplicated the setup over there. They have completely the same setup. But instead of having a lot of instruments, they have one of the sampling line that goes to the mouse exposure chamber. So this is what the chamber looks like. On the top, we have a dome-like cover. And from the top, the smoke goes to the mice. And there are like 7 mice that are like placed in this chamber. And this is the exhaust. And after the experiments are performed, so mice are exposed for 2 hours every day. And we have like short-term study that's like after a week, and then long-term study like after a month or like after 2 months. Over there, like students dissect and understand the effect on the lungs and heart of the mice. That's what is happening at Northeastern University presently, like right now. Okay, moving on to what is the further step of this research. We will be assessing the PPEs that firefighters use. How effective are they to control the particulate matter? And are they like really efficient or they're not efficient? So we will be doing this study, like the later steps of this research. And we'll also study like the fuels from the California that we will be doing. And we'll also try to increase the number of effluents, because we know we are getting hydrocarbons, but what are those hydrocarbons? So these are the next steps in the research. Yep, at last, I would like to thank all the people who are involved in this study. And this study is supported by FEMA and with the assistance of Firefighter Grant Project. Thank you.
Great, thank you so much, Priya. So we have another question in the Q&A, and also if people have questions post-talk, we'll leave the chat open for a couple minutes before we have our next talk. But so again, Joseph is asking, do you have an opinion on usefulness of homeowners installing low-cost real-time smoke monitors, such as Purple Air?
I think, yes, it will be useful to install them because you get to know how much are the particles in the air around. So it's good to know, but it's better to have like the filters, air purifiers, especially during the fire seasons.
Great, thank you. We'll wait to see if maybe another minute, if anyone has any last questions. So what are the main components of California wildfires, I guess? Like what plants are you hoping to look at?
I'm just gonna like do more research research on the fuel, which fuel are the worst ones, and like what's the good moisture level. Because if we understand like which is the better moisture level, we can tell the field managers to burn the fuel like during the prescribed fires, to burn the fuel at this moisture content, like, you know, whether it should be like pretty dry or like absolutely dry or like live. What's the good state to burn that? So that's like what we will be going into.
Cool. I kind of forgot about prescribed fires, and you said control the moisture content of a fire. I was thinking of wildfires where that's not quite something you're able to do, but prescribed fires is a cool application too.
Yeah.
All right. It looks like there aren't currently any questions coming in, so we'll switch over to Mallika's chat. Mallika, if you could share your screen. But as a reminder, we will keep the chat and Q&A open during this talk. Priya's free to answer your questions, but we'll also stick around for a few minutes after Mallika's talk. And so for any final questions for either of these talks. So yeah, we have Mallika here today, who's a fifth-year PhD student in the Materials Science Department. Before that, she did her undergrad in engineering physics at Cornell. And then before that, she was homeschooled in San Jose and here in California and Bangalore, India. So she's been all over the place. But currently, she now works at Berkeley to study and develop wearable sensors to detect body health that don't require invasive blood tests. So I think she's going to talk to us today about how we can potentially use sweat to monitor our health. So please take it away, Mallika.
Thanks so much for the introduction, Madison. Can everyone hear me and see my screen?
Yes, you look great.
Okay, excellent. I've been having trouble with my internet, so I'm actually going to turn off my video to make sure this goes simply. So just want to say hi so everyone here can see my face before I disappear behind the black box. Okay. So thanks so much for the introduction. Like Madison said, I'm Mallika. I'm a fifth-year PhD student in the Material Science Department at Berkeley, but my research is actually in the Electrical Engineering Department. So I work at the intersection of these two fields on wearable sweat sensors. My talk today is called Sweat It Out. What can molecules in sweat tell us about our health? Sorry, Zoom problems. Okay, there you go. So before getting into the specifics of sweat, let's first take a step back and ask, why is there growing interest in wearable sensors? And the answer is that there are many limitations to the way that we conventionally perform diagnostics and health monitoring. So traditionally, someone who wants a health checkup would go into a hospital where a trained specialist, maybe a nurse or a doctor, would make an invasive, often painful blood draw. That blood sample would then be processed with bulky lab equipment by another trained specialist, and then sometime later, could be hours or days, you would get that blood sample result back. So overall, this is a lengthy and often expensive procedure, and the information you get is discrete. It's really only a single snapshot of the state of your body at the time that that blood sample was taken. In contrast, wearable sensors are systems that are worn on the body to make measurements automatically, in situ, and immediately. So you no longer need trained specialists, you don't need bulky instruments, and you can now get health information in seconds or minutes rather than in days. Even further, these measurements can be made continuously, so you can track how your health changes over time and detect health conditions as soon as they occur, or based on the data, maybe even predict them ahead of time. So this is kind of the big vision of wearable sensors in general. There are many wearables that are common today, including smartwatches that can measure heart rate or breathing patterns, but very few are actually able to measure chemicals in the body. There's this outstanding need to access chemicals conveniently without puncturing the skin or relying on blood. And that's where sweat comes in. So how does sweat provide chemical information about the body? Let's start by considering how sweat is produced. This is a schematic of a sweat gland located in the upper layers of skin. And what's not shown here is that this gland is actually surrounded by blood vessels. So along this whole length of the gland, there are capillaries kind of twining around this duct. When sweating is initiated, water is pumped into the secretory coil at the base of the sweat gland, and then it travels up the dermal duct to the skin surface where it's secreted as sweat. And during this journey, a variety of molecules migrate into sweat from the surrounding cells and blood vessels. So this includes molecules like ions, maybe sodium and potassium, metabolites like glucose and lactate, various acids, hormones, proteins, and even DNA. So overall, sweat has this very rich composition of chemicals that are sourced from deeper in the body. And this creates opportunities for using sweat to probe body chemistry without needing to directly and invasively access blood. So we've seen that sweat contains a bunch of different molecules, but what does sweat testing with wearable sensors actually tell us? And there are two main application classes, medical versus athletic. And within these, there are certain established applications where sweat has historically been a gold standard, but has traditionally been collected and tested off body in a lab. Wearable sensors instead can make these tests more rapid and even remote. So instead of collecting sweat samples and testing them remotely, and instead of having all of those time delays, wearable sweat sensors can make these measurements much more immediate and at the point of sweat secretion. So examples of an established medical application include diagnosing cystic fibrosis. And this is a respiratory disorder that is also marked by patients having unusually salty sweat. So this can be diagnosed with wearable chloride sensors. An athletic application that is also established is doing sweat tests for doping control. So in this case, an athlete would wear a sweat patch. Traditionally, that patch would be worn for maybe 24 hours. It would collect various molecules that are excreted in sweat, including illicit drugs. And you would then take that patch to a lab to be rehydrated and analyzed. And in this case, we could instead have wearable sweat sensors that are making those drug measurements on body, so on the skin surface directly. Beyond these examples, there are exploratory applications of sweat. And these are applications that need to be further studied. So they're kind of opportunities that having wearable sensors opens up, but these are ideas for how we can kind of continue to explore what sweat could be used for. So for example, type 2 diabetes is traditionally diagnosed using finger stick blood glucose tests. But we know that sweat contains glucose. So sweat might be a viable alternative fluid to test to diagnose diabetes. On the athletic side, dehydration is a potential application. This is a challenging body condition to diagnose in athletes. One way it's traditionally done is to consider the color of an athlete's urine, but this isn't very quantitative and it requires exercise to be stopped, and by nature, it's a discrete measurement. So instead, the hope is that we could measure the amount of sweat fluid loss that is occurring during exercise. We could measure this in real time and use that to indicate when an athlete is becoming dehydrated. So not just after exercise, after the fact, but to really see how dehydration is happening, when it starts, how it progresses, and use this as a way to potentially preemptively prevent dehydration from happening at all. So these are some examples of established and exploratory applications of sweat sensing. But given its rich composition, sweat tests might be relevant to a whole bunch of other applications. And this is based on molecules that exist in blood that are traditionally used to diagnose these various conditions or used for various medical applications. Those molecules are also found in sweat. So it creates these opportunities for using sweat to supplant blood tests. One thing I want to emphasize here is that you don't need to exercise to sweat. For athletic applications, sweat is, of course, copiously produced and available, but we definitely cannot expect to exercise anytime we want to do a health test. And for certain groups of individuals, including patients or the elderly, exercising may not even be an option. Happily, there are alternatives. So we can do a process of local sweat stimulation, which is called iontophoresis. And this is a process whereby a sweat stimulating drug called pilocarpine is transdermally delivered non-invasively using topical, so using current application on the skin surface to drive pilocarpine under the skin, locally stimulate the sweat glands, and that produces local sweat. So we have this technique of stimulating sweating without needing to exercise. And this can be done while an individual is just sitting at rest, is sedentary. Beyond this, we are also actually sweating naturally at all times. So even right now, while we're sitting, either giving this talk or listening to this talk, our bodies are sweating, and we keep doing this to finely control our core temperature. This sweat is secreted at very low rates and it evaporates quickly, so we don't notice it. But with the right sensor design, you can actually capture that amount of natural sweat for analysis. And this is a really neat video. I found it on YouTube. It shows the process of natural sweat secretion. So this is a person, they're just at rest, they're not exercising, and you can see, we go really close in, you can see the process of natural sweat secretion without needing exercise to stimulate it. This looks a little bit disturbing, but it's also really impressive that we can actually visualize this. And this is showing that sweat is actually always available. As long as we have the right devices, we can actually capture this.
Mallika, may I ask a question? Sweat is always available, but is it always, like, are these sweats the same type? Would they have the same concentration of a given molecule that's in your system? Or would natural sweating be very different from exercise sweat?
That's an excellent question. And yes, all of these types of sweating will be very different. So the way that you stimulate sweating can impact the composition. If you're exercising, you can imagine that your body is in a very out of equilibrium physiological state. So the concentrations of molecules that you'll be seeing in sweat are reflecting that dynamic state as opposed to your resting state. So not only would you not want to exercise for medical applications just because it's inconvenient, but you would really be probing a very different state when your body is at rest versus when it's exercising. And beyond that, also the composition of sweat can vary at different body sites. So there's a lot of complexity here about the type of sweat that we want to target and where on the body we want to target.
Cool, thanks.
Yeah, excellent question. Okay. Okay, so we've seen that we can access sweat at different body sites, and we know that it has a rich composition. But before I get into the details of how we can actually make chemical measurements in sweat, I wanted to very quickly just show what sweat sensors actually look like. So this is a sampling of sensors produced by my group and by other groups around the country, and it gives a flavor of the sort of structures that sweat sensors can have. So they can be packaged as bands, as finger cots or gloves, as tattoos that are laminated directly onto the skin, as patches of all different forms and all different body locations. And I'm actually going to just focus on this one here, just because it contains many of the characteristic elements that are common across sweat sensors, and because this is kind of one of the prototypes that our group developed. Okay, so this is where I'll describe the anatomy of a typical sweat sensor. There are three main components, and these are microfluidics, embedded chemical sensors, and electronics to actually process the signals that we're measuring. Microfluidics are essentially narrow tubes that accommodate very small amounts of fluid. And this is important to have because it lets us trap sweat to prevent evaporation, to remove sweat from the skin surface so it's not contaminated by any other kind of secretions on the skin. And it lets us control the movement of sweat over the chemical sensors that are embedded in this device. So as an example of how this works, we have in this patch shown here, we have this spiraling microfluidic, and it has this central opening well. And that opening well interfaces with skin. So that's where sweat glands in that area that are secreting, that's where they produce sweat, and it moves up into this central opening and is pushed into the device for measurement. And in this cross section here, you can see how that would work. We have this kind of increasing volume of sweat. It fills up this collection well, and once it does, it gets pushed into this enclosed microfluidic. Okay, so that's the first component. The second component is the chemical sensors. And in this case, they're located here in the central well, kind of close to where the freshest sweat is being secreted. And here I'm just showing an exploded view of what those look like. So this microfluidic layer would essentially spiral over these electrodes, and this molecule detection area would be exposed to your sweat. So these chemical sensors are constructed as follows. They are a bunch of metal layers, metal electrodes deposited on a flexible backing, on top of which in this molecule detection area, we deposit various chemicals and materials that selectively interact with the molecule we're trying to measure. So as an example of how this works, here's a kind of rough cross-section that we can pretend represents a glucose sensor. So this is our kind of flexible backing. This gold cross-sectional area would be a cross-section of our underlying metal electrode, which might be gold. And on top of this, we've deposited various materials that essentially attract these biorecognition molecules. So in this case, this is what I'm showing as kind of blue dots. That would be, for this glucose sensor, this would be the glucose oxidase enzyme, which is an enzyme that selectively interacts with glucose and not with other molecules in sweat. And that's very important because that selective interaction is what makes this a sensor. It cannot be a sensor if it is not selective. So when this sensor is immersed in sweat, glucose in sweat, which I've kind of schematized as little red dots, glucose in sweat interacts with the sensing surface. It actually diffuses into the chemical materials of the sensor, and it actually, the glucose molecules interact with glucose oxidase, and they react and produce electrons. They produce a measurable current. And this current is what we measure in this device and calibrate back into a concentration reading. So the more glucose that's present in sweat, the more electrons we get out of our electrode, the higher the current that we're measuring, and the bigger the sensor. And we can calibrate that bigger sensor into a bigger concentration reading. This current flowing out of the electrode is measured by the third component, which are the electronics of this device. And this set of, over here, it's just this flexible printed circuit board. It has a lot of complex circuitry, but essentially the role of this device is to process the signals, remove noise, and wirelessly transmit them to a computer or a smartphone where those signals can be read off in real time and can be calibrated into a glucose concentration measurement. So this is kind of the anatomy of a typical microfluidic sweat sensor with embedded chemical sensing components. So now that we have a better sense of the anatomy of a sweat sensor, we can go back to the different sensors I showed before, and we now have a better sense of how these work. So you can see if you look at all of these different devices, not all of them have all three components. The ones at the top here, for example, they have electronic components here. This one has electronic components. What you see here kind of as a tattoo on the skin, those are electrodes, functionalized electrodes. So these are sensors directly laminated on the skin. They don't have microfluidics. And in this case as well, that sensing patch with the electrodes doesn't have microfluidics. In this case, these are just the electrodes. We're seeing the back of it. They're actually within this finger cot interfacing with skin. You would attach electronics to this, to the ends of these electrodes to make your measurements. But again, we don't have microfluidics in any of this. So what this means is that when we don't have microfluidics, these sensors are more vulnerable to sweat evaporation, which means we have to be careful when we try and interpret the measurements that we're making. We might see artificially high concentration readings just because we're actually losing sweat volume. And beyond this, microfluidics are very important because they help carry old sweat away from the chemical sensors so that new sweat can take its place. And that really allows us to make updated, refreshed, real-time composition measurements in the freshest sweat. So without microfluidics, these sensors are really most appropriate for doing single point rapid measurements.
Mallika, I have a question about the microfluidics.
Mm-hmm.
Is there a certain type of shape that's better for sweat sensing, or do people just generally design whatever is the optimal design based on their overall structure of the sensor?
Yeah, it's a good question. So in this case, the reason we have a spiral is it just helps us consolidate the sensor footprint. So we can hold a reasonably large amount of volume in this device without it extending far out in different directions. Mm-hmm. But we've also have devices that have a more serpentine design. Mm-hmm. And you can kind of have fun with this. I mean, I imagine that it's good to optimize it. In this case, it works well to just have a spiral. But mm-hmm. there are some pretty funky flower-like microfluidics that people use and make really cute sensors as well.
Oh, okay. Cool. Thank you.
Mm-hmm. There's a lot of optimization that goes into designing the dimensions of the channel that can impact the rate of fluid flow over your sensing electrodes, things like that. So there's a lot of work that goes behind it. But for proof of concept, I've seen a lot of different types of designs. So microfluidics are very important to prevent evaporation, to make updated sweat measurements. And you can see in these two cases, we actually have these microfluidics. Both are spiraling formats in this case. But overall, sensors can take on a whole different number of shapes, of formats. Some have microfluidics, some don't. This device uses an entirely different sensing scheme. Instead of doing these sort of current measurements, it uses dyes that change color when they interact with their target molecule. So this is, you can do optical or even visual readout. The intensity of the color can be related back to a concentration measurement. And that's really nice. You don't actually need electronics attached to these devices. You can see it's just in itself, this is the complete sensing patch. The disadvantage is that it's hard to make real-time measurements because once the dye interacts with its molecule, it doesn't release those molecules. So you can't make updated measurements. So there's a lot of choices that go behind making these sensors, behind designing the format of the microfluidics and choosing your measurement scheme. But overall, sensors can be designed in varying formats. They can be designed for different body size. They can be functionalized towards different molecules, and they can be targeting continuous monitoring or more geared towards single. So there's a lot of variety for the sweat sensors that have been developed so far. So I've shown many types of sweat sensors, and I've discussed that there's a lot of molecules that you can detect in sweat. So why aren't these sensors broadly available yet for health testing? The answer is that even though we have the technology to measure molecules in sweat, and of course it needs to mature further, we need to make sure that our sensors are very reliable, but a lot of the innovation has already been done. So why aren't these sensors broadly available? And the answer is that even though we have the technology, we don't always know how to interpret our measurements. So for example, commercial tests have interpretable ranges. For example, if you're trying to diagnose type 2 diabetes, you can prick your finger, get a drop of blood, and use a glucometer to make a blood glucose concentration measurement. And based on the range of glucose levels that you see, you can kind of get a sense of your diagnosis. So if you're making these measurements in the morning when you're fasting before you've eaten breakfast, if you see that you have a blood glucose reading less than 100, you're likely to not have diabetes. And of course, you have to repeat this test. So this is kind of at a high level, this is what you would expect. If you have a reading between 100 and 125, you might be pre-diabetic. And if your reading is over 125, there's a high chance that you have type 2 diabetes. So these are well-defined ranges, and they are globally applicable across individuals. And the big question is, can we develop similar ranges for sweat? And we need to do that to be able to use sweat as a viable alternative for blood. So towards this goal, we did a population study across a group of diabetic and non-diabetic individuals, comparing their fasting blood glucose and sweat glucose levels. So on the horizontal axis, we have blood glucose and we have sweat glucose on the vertical axis. And blue points represent healthy or non-diabetic subjects, and red points represent diabetic subjects. And what you can see here is that along the horizontal axis, there's a clear segregation of the healthy and the diabetic cohorts. And this makes sense because blood glucose is known to be a good way to differentiate between these two populations and to test whether you are healthy or diabetic. So we see that healthy individuals cluster towards the lower blood glucose end, diabetic individuals cluster towards the high blood glucose end. If instead we look on the vertical axis, we don't see this type of segregation. So for example, if we look at 70 micromolar sweat glucose, we draw a horizontal line, we see that that line goes through healthy cohorts as well as diabetic cohorts, which means we can't really differentiate based on sweat glucose whether an individual is more likely to be healthy or diabetic. And what this is telling us is that unlike blood glucose, sweat glucose tests alone, they cannot simply and globally be used to diagnose type 2 diabetes. What we can instead do, and this is work done by another group in the country, what can instead be done is to consider how a single person's blood and sweat glucose compare over time. So instead of doing a population study across a number of different individuals, we do a longitudinal study where we're looking at temporal changes in blood and sweat and how they compare for one individual over time. You can see here that the trends track each other. So we have the red points represent blood glucose measurements, the black points represent sweat glucose measurements. This is monitored over more than 7 hours and over the course of taking 2 meals. So this dotted line marks the time when breakfast is consumed. This is when lunch is consumed. And we see that these that blood glucose and sweat glucose tend to roughly track each other. So blood glucose goes up after you eat. That makes sense. You have energy in your body, you have glucose released in your bloodstream, and then it goes up again after lunch, and that similarly holds true for sweat glucose. They roughly, I mean, there's some variation, they don't exactly track, but we see that these rough trends exist. And this creates hope that for an individual, we might still be able to use sweat glucose to indicate what blood glucose values would be. But the relation between the two will differ from person to person. That's what we see with this population study. We're not able to find a universal correlation, but there seems to be a correlation for an individual over time. And this is okay, because a huge advantage of wearable sensors is it allows for this type of continuous monitoring. It allows for personalized correlations and algorithms to be developed such that we can interpret sweat measurements on a person-to-person basis without needing a single universal method. But this is ongoing work. It requires a lot of data to be collected and to be studied to develop these personalized interpretations so we can better understand how sweat measurements relate to blood or to physiological state across people and over time. And once we do this, once we establish these correlations and a better understanding of how sweat measurements can be interpreted for an individual or across a population, that's when we have a stronger basis for promoting sweat sensors beyond a lab or real world use. So in summary, this is a rough roadmap of wearable sweat sensing and all of the work that's involved and that is yet to be done. So at the start, there's a lot of platform development, kind of putting together the different components, working on the electronics, working on networks of sensors, wireless data transmission schemes, things like that. Another goal is just developing the actual sensors, so figuring out the chemistries and the materials that can be used to functionalize metal electrodes to make them selective for different target molecules. And then once you have these in place, you can perform these sort of correlation studies that I showed in the previous slide for glucose. So you can look at how sweat parameters change over time and with body state and use that to kind of identify individual and universal correlations that can help us interpret sweat signals into kind of actionable health information. And once you do this, you can also mass produce sensors. What I'm showing here is something called roll-to-roll processing and fabrication. It's a way to mass produce printed sensing components and microfluidics. And a lot of this work is actually back and forth. So you need to mass produce sensors in order to have enough devices to actually perform correlation studies. And as you do more correlation studies, you might discover that there's certain interdependencies between different sweat parameters. For example, we know that your sweating rate could impact the concentration you measure. We know that sweat pH could impact the glucose sensor signal we get. So along the way, you might have to develop new sensors and do what we call multiplexed monitoring. So monitor a number of parameters simultaneously to create more interpretable data. And ultimately, once you kind of iterate through all of these steps and you develop significant correlations enough to be able to interpret sweat measurements, then you can start promoting sensors beyond a lab for real world use as a viable alternative to lab tests. That's all I have. I have contact information here. I have a link to my group website. There's a lot of publications there, but if you do Control F and type in sweat, you'll see all of the relevant ones. And I can take any more questions if there are any.
Great, thank you so much, Mallika. That was a really cool talk. We do have one question currently in the Q&A that asks, what is the difference between microfluidics and microchip sensor technology? Do they both have potential use as sweat sensors?
Yeah, that's a good question. So microchip sensor technology could refer to a bunch of different things. And the micro there is kind of referring to the scale of devices. It could refer to how consolidated your electronics are. That's sometimes what we think of when we're talking about chips. Microfluidics are really focusing on kind of structures that allow us to control microliters of fluid. So they're really a way to handle small fluid volumes. And definitely both microfluidics and microchip technology kind of come together when we're making wearable devices.
Cool. And then also the question, could these devices have potential use for early indicator of heart disease?
Yeah, they could be. And that's there's kind of wearable sensors that don't look at sweat, that actually look at physical indicators like your heart rate, variations in heart rate variability is called, and that can be an indicator of cardiac health. And we're hoping that we can make certain chemical measurements in sweat that are traditionally made in blood to also give a better sense of your cardiac health. And that's work that still needs to be done. So the next step for a lot of these sensors is really to perform clinical trials to see how sweat parameters relate to different health conditions.
So obviously you touched on a few of them, but do you have any idea, or what would you say are kind of like the main applications currently of sweat sensors? I mean, you talked about like athletic dehydration and doping and stuff, but for the general population, maybe like what are the main what main things are we sensing currently or want?
Yeah, so the ones that we know that there are strong correlations are, one would be cystic fibrosis. We know that sweat chloride tests can definitively help diagnose that. We know that sweat ethanol levels correlate very strongly to breath ethanol levels. So you can use it as kind of a measure of alcohol in your body and the rate of alcohol absorption. You can measure heavy metals. Sweat so far actually is very good at measuring or detecting things that are called xenobiotics. So they're not natively found in your body, but come in through your food or through environmental exposure. So drugs, alcohol, heavy metals that you might be exposed to through bad drinking water, all of those are commonly excreted in sweat. So sweat is a very good contender for detecting that kind of thing. And if we want to use sweat to measure endogenously produced molecules like glucose, things that are used by the body and have more complex absorption and metabolism pathways, there we still need to do more work.
Very interesting. All right, I think we have a little bit of time. If there are any final questions in the chat or Q&A, we can wait for a minute or so.
On behalf of the library, I do want to thank both of our presenters tonight. Priya and Mallika, you have done such a great job, such great topics. You know, I think that fires are something we're all interested in, health topics. It's just amazing to see the research that's happening there. Thank you.
Thanks, Kelsey.
Yeah, and if you guys in the audience are particularly interested in either of these topics, I mean, these talks will, of course, have been recorded and will be published, but we also have resources given to us by both of our speakers that we will share with you. We'll send out in both a tweet and Facebook link, and I think potentially in a follow-up email, but you can always follow or find those resources linked on our webpage as well. All right. It looks like we might be wrapping up.
All right. I think that closes us out for the meeting. All right. Thank you so much. Have a good night.